morning and healthy sweetie day for all from around the world, and especially who work on this conference, the 8th International Conference of Biotechnology, Environmental and Engineering Science in Stockholm, Sweden. My paper and the title Glyphosate Degradation by 4 plant growth promoting bacteria detected HPLC assay, presented by Nibal Mousa, Agriculture Engineer Manager, Master Degree in Biotechnology from USA in 2011 and Master Degree Agrobioengineering in Poland 2018. Work in Iraqi Ministry of Science and Technology, Baghdad, Iraq. Abstract. The best eco-friendly, cost-effective and efficacious nature is pesticides bioremediation. For plant growth promoting bacteria used in glyphosate biodegradation, Bacillus megater, Bacillus subtilis, Rhizobium and Azotobacter were grown on mineral salt media for 2 to 60 days with the glyphosate concentration 5, 10, 15, 20 and 25 ppm. The glyphosate degradation ratio showed 70.9 to 91% for 25 ppm in 60 days for Bacillus megateria and Bacillus subtilis respectively. The best Rhizobium degradation ratio was for 25 and 2 to 60 days reached 96.24 to 96.25%, while Azotobacter best degradation ratio was for 25 ppm reached 54.16% at 60 days. The HPLC residues for each Bacillus megater at 25 ppm 30 to 60 days reached 89 to 91%, and Bacillus subtilis 15 ppm to 14 days reached 90.3%. Rhizobium species at 25 ppm to 30-60 days reached 93 to 78%, while Azotobacterium the 25 ppm and 30 to 60 days reached 82 to 79% respectively. The conclusion that all bacteria types tend to the 25 ppm for 30 days was Rhizobium 93%, while for 60 days was the Bacillus megaterium 91%. Improved glyphosate biodegradation as a source for phosphorus and suggest could be well exploited for bioremediation or biodegradation of glyphosate contaminated soil or water for the period reached 30 to 60 days. Introduction. The pesticides are environmental pollution to the leaching and to the soil water body. Some studies show low leaching risk glyphosate due to strong sorption characteristics, while other studies indicated a leaching risk into the deeper soil layer that could end up in the surface water and the groundwater. The sorption level condition can be changed according to the organic matter, the soil mineral type, contains phosphorus, soil acidity range, pH, redox condition of soil beside rainfall and poor state of soils. Glyphosate degradation microorganism mechanism in soil also produce amino methylphosphoric acid and compound saccharine production. The microorganism degradation mechanism break down the soil bacteria population and their enzyme supply NPC, nitrogen, phosphorus, carbon source. Overall, soil type, high temperature and topographic glyphosate sorption onto soil particles led to decrease its degradation. If it has soft microorganism will still degradation it. Thus, the study aimed to demonstrate and investigate the power of glyphosate degradation by 4 plant growth promoting bacteria and detect its residues under laboratory conditions and HPLC assay in different periods. Minerals and methods. Chemical and reagents. Glyphosate supply from Iraqi market. Chemicals and reagents were available in laboratories of Remediation, Pollutants, Sensor, Iraqi Ministry of Science and Technology. Bacteria was grown and mineral salts media as shown in table 1 with glyphosate 5, 10, 15, 20, 25 ppm and without glyphosate. The phosphorus examined their growth and degradation. Plant growth promoting bacteria were Bacillus megaterium, Bacillus subtilis, Rhizobium, and Azotobacter were kept under incubation in the laboratories of Remediation Pollutant Center with none of their biomorphological and biological tests recultured before used on mineral salts medium. Plant growth promoting bacteria growth were measured by spectrophotometer at 600 nanometer lambda measured bacteria ability growth on mineral salts with and without glyphosate. The biodegradation ratio measured from 48 hours to 60 days for glyphosate degradation ratio measured by spectrophotometer under 600 nanometer lambda depending on bacterial growth to measure according to the equation. The biodegradation ratio equal 1 minus A1 in the sample of glyphosate and A0 sample with non-glyphosate. HPLC assay. Glyphosate extracted from mineral salt media samples of 30 to 60 days. In the tube added 3 mol of mineral salts ethyl acetate, shaken twice, timer applied the mixture of suspension where centrifuge 3000 rpm to 10 minutes, filtered and dried them. Filtration through glass fiber paper. Each of ethyl acetate fraction was analyzed by HPLC condition. HPLC condition as in table to the equation for calculating the final glyphosate concentration as shown here. Results and discussion. A plant growth promoting bacteria growth, the best growth were Bacillus megaterium, the 5 and the 25 ppm for 60 days show 0.164 and 0.167 respectively. Bacillus subtilis, the 10 to 15 ppm for 3 days show 0.2 and 0.19 respectively. Rhizobium, the 25 ppm for 7 days show 0.19. Azotobacter, the 25 ppm reached 0.222 and 0.163 in 30 and 60 days as in figure 1 for each one. HPLC assay where Bacillus megaterium was 25 ppm reached 9.013 and 3.8 for 30 and 60 days. Bacillus subtilis was 25 ppm reached 6.6 and 6.8 for both 30 and 60 days. Rhizobium were 25 ppm 2.81 mg per liter for 30 days, while for 60 days reached 25 ppm reached 12.6 mg per liter. Azotobacter also for 30 and 60 days for concentration 25 ppm reached 3.1 and 4.2 mg as in figure 3. The mainly factor explanation microorganism pesticide degradation is based on enzymes' effect on the texture of them that led to substrates in soil depending on microbial population size and not all microbes can metabolism. These products tend to persist, suggest use the tetrachlorophenols organophosphorus pesticides as a carbon and energy source. Flavobacterium species isolated from soil and OP pesticides contact generated phosphotiesterase enzyme resulting as a metabolic pathways degradation. The co-metabolism microorganism can be the bacteria enzyme converted substrate to organic products that will utilize in energy protection. Also that substrate transfer to compound inhibited mineralization activity of enzymes, Bacillus megaterium degradation ability, each of chloropyrifos 600 ppm for 240 hours, atrazine 50 mg per kilogram for 168 hours, and monocryptose reached 81.99 and 83% respectively. The results of the presented study suggest that the PGPB Plant growth promoting bacteria are capable of growing in media utilizing pesticides as a phosphorus and or carbon source that using the bioremediation of pesticides contaminated soil. Conclusion: the best results of plant growth promoting bacteria shown in the growth percentage Bacillus megatherium, the 5 and the 25 ppm for 60 days, showed 0.164 and 0.167 respectively. Bacillus subtilis in 10 and 15 ppm for 3 days showed 0.2 and 0.90 respectively. Rhizobium, the 25 ppm 7 days show 0.19 and Azotobacterium at the 25 ppm show 0.222 to the 0.163 in both 30 and 60 days. The biodegradation ratio showed in Bacillus megatherium 25 ppm for 60 days reached 17.90%. Bacillus subtilis 15 ppm to 40 days reached 19.32%. Rhizobium species 25 ppm showed in 2 and 60 days reached both 19.6.2%, and Azotobacterium 25 ppm reached 54% in 60 days. The HPLC assay for detect glyphosate ratios showed Bacillus megatherium was 25 ppm reached 9 and 3.8 for 30 and 60 days. Respectively, Bacillus subtilis was 25 ppm for 6.6 and 6.8 for 30 and 60 days respectively. Rhizobium was 25 ppm for 2.81 mg for liter 30 days and 25 ppm reached 12.60 mg for 60 days, and Azotobacterium was 25 ppm for 30 and 60 days shows 3.1 and 4.2 mg per liter. Finally, thanks for attention and wish to meet you in another conference.